Dr. Sage here to continue our discussion of genetic analysis and genetic engineering. In today's video, we're going to discuss genetic treatments introducing DNA into the body. By the end of this video, you should be able to differentiate between somatic and germline gene therapy and describe miRNAs and ways in which their discovery can impact human disease. For certain diseases, the disease phenotype is due to the lack of a single specific protein. For example, type 1 diabetes is caused by the lack of insulin. Insulin was first provided from the pancreas of pigs or cows. Recombinant human insulin was the first genetically engineered drug approved for humans. Gene therapy is the repair or correction of a faulty gene in humans suffering from a fatal or debilitating disease. A benefit of gene therapy is that it's a permanent cure of the physiological dysfunction by repairing the genetic defect. In gene therapy, a normal gene is cloned into a retrovirus or adenovirus vector. Tissues are removed from the patient and incubated with a genetically modified virus to transfect them with the normal gene. Transfected cells are then reintroduced into the patient's body. Naked DNA or a virus vector is directly introduced into the patient's tissues. Experimental and clinical trials of somatic cell gene therapy were carried out on human volunteers with a particular genetic condition. Most target cancer, single gene defects, and infections. Most gene deliveries are carried out by virus vectors, and early trials were hampered by safety issues in which the virus used as a delivery vehicle caused malignancies. In germline gene therapy, genes are inserted into an egg, sperm, or early embryo. The new gene is present in all cells of the individual, so the therapeutic gene is heritable. Diseases resulting from overexpression of a protein include Alzheimer's disease, viral diseases, many cancers, and sometimes this occurs due to a malfunctioning microRNA or miRNA. We can also use small RNAs as medicine. Cells use a variety of microRNAs to silence gene expression, so we can use microRNAs to fight diseases. Introducing specific microRNAs into small cell lung cancers in mice block tumor growth. MicroRNAs cause regression of liver cancers in mice. We also can inhibit microRNAs to fight diseases. Inhibiting specific microRNAs in mice stop the growth of ovarian and breast cancers. Inhibiting microRNAs in African green monkeys lower their plasma cholesterol levels. And multiplication of hepatitis C in chimpanzees was inhibited by shutting down one class of microRNAs. In the first successful human application of interfering RNAs, healthy human volunteers were given a nasal spray containing microRNA designed to silence a gene from RSV, which is a virus that causes mild cold-like symptoms. Volunteers were then infected with RSV and used the spray for 5 days. 44% of those using the miRNA were infected compared to 71% of control subjects. Among those that were infected, symptoms were significantly reduced. Alright, this was your brief introduction to genetic treatments introducing DNA into the body. Until next time, this has been Dr. Sage.